Hello and welcome to Professor Gadgil's GPAT Academy. Today we will discuss about the pharmacology and chemistry of penicillins. Penicillins are beta-lactam antibiotics originally derived from the fungus Penicillium chrysogenum. They are bactericidal and act by inhibiting bacterial cell wall synthesis. Penicillin binds to penicillin binding proteins, the enzymes involved in the peptidoglycan cross-linking. They inhibit the transpeptidase, the enzyme that cross-links peptidoglycan chains in the bacterial cell wall. This leads to defective cell wall and results in osmotic lysis of the bacteria. They also activate the autolysins, that is murin hydrolases, further degrading the cell wall. They are bactericidal only against actively dividing bacteria, that is cell wall synthesis occurs during division. Now we will discuss the classification and spectrum. First class is the natural penicillins. Examples include penicillin G, penicillin V. They are narrow spectrum. They target the gram-positive cocci, Treponema pallidum, Clostridium, Actinomyces. Penicillin G is acid labile, while the penicillin V is acid stable. Next are the aminopenicillins. Examples include ampicillin and amoxicillin. They are extended spectrum penicillins. They also inhibit some gram-negatives like the E. coli and H. influenzae. Amoxicillin has better oral absorption. Third is the anti-staphylococcal or the Penicillinase-resistants. They are methicillin, oxacillin, cloxacillin, dicloxacillin, and nafcillin. They are resistant to staphylococcal beta-lactamase and used for MSSA, that is methicillin-sensitive Staph aureus. Next is the antipseudomonal, that is carboxypenicillins and uridopenicillins. Examples include carbenicillin, ticarcillin, piperacillin. They are broader gram-negative, including the Pseudomonas. They are often combined with the beta-lactamase inhibitors. Last are the beta-lactamase inhibitor combinations. Examples include amoxicillin plus clavulanic acid, ampicillin plus sulbactam, and the piperacillin plus tazobactam. They are the inhibitors to protect penicillin from the beta-lactamase enzymes. Next we will discuss the pharmacokinetic profile. Absorption. The penicillin G is acid labile, hence it is given parenteral, while the penicillin V is acid stable and hence oral administration. Amoxicillin is well absorbed and food does not affect the absorption. Distribution. They are widely distributed. They have the poor CNS penetration except in the inflamed meninges. They have high protein binding. Next is the metabolism. They have minimal hepatic, mostly are excreted unchanged by kidney via the tubular secretion. Probenecid blocks the renal tubular secretion and increases the penicillin levels and is sometimes used to prolong the action. Excretion is via renal except for the nafcillin and oxacillin. They have the significant biliary excretion. Dose adjustment is required in the renal failure. Next we will discuss the resistance mechanism. First is the beta-lactamase production, which is the most common. Staphylococci, E. coli, H. influenzae, and the penicillinase hydrolyzes the beta-lactam ring. It is the most common defense. The enzymes physically hydrolyze the beta-lactam ring, yielding the inactive penicilloic acid. Next is the altered target PBPs. The methicillin-resistant S. aureus expresses PBP2A, an altered receptor which fundamentally lacks the affinity for the penicillins. Third is the decreased permeability. The gram-negative bacteria possesses a restrictive outer membrane and narrow poring channels that physically impede large molecules from entering. And last is the efflux pump, the active transport membrane systems that continuously and aggressively pump the penicillin molecule back out of the bacterial cell. Next we will discuss the important adverse effects of penicillins. First is the hypersensitivity, which is most common cause of drug allergy. Rash, urticaria, and anaphylaxis are some of its symptoms. Cross-allergy among all penicillins can occur. The possible cross-reactivity with cephalosporins is seen. Next is the Jarisch-Herxheimer reaction. It is due to rapid killing of spirochetes in the syphilis, releasing the endotoxins. Next is the GI upset, diarrhea, nausea. Ampicillin causes pseudomembranous colitis. Next is the CNS toxicity. Seizures at very high doses or in renal failure can occur. Next is the hematologic, that is hemolytic anemia and neutropenia after prolonged use is reported. Next is the electrolyte imbalance. Penicillin G potassium can cause hyperkalemia if high dose is given. Ticarcicillin has high sodium load. Next, we will discuss the medicinal chemistry of penicillins. The core nucleus is the 6-amino penicillinic acid or the 6-APA. The 6-APA consists of the beta-lactam ring, which is a 4-membered cyclic amide, and a thiazolidine ring, which is a 5-membered sulfur-containing ring. The beta-lactam ring is fused to the thiazolidine ring to form the bicyclic system. The side chain R is attached to the 6-amino group, which determines the spectrum, stability, and the pharmacokinetics. There are certain structural features which are essential for the activity. The beta-lactam ring is essential for the antibacterial activity. It is highly strained and reacts with the PBP active site serine. The free carboxylic acid on the thiazolidine is essential for binding to PBP. It is usually as a sodium or the potassium salt. The cis stereochemistry at fusion is required for activity. The trans is inactive. The acyl amino side chain at C6 determines the spectrum, acid stability, and the beta-lactamase resistant. Sulfur atom in thiazolidine is essential. Oxidation to sulfone destroys the activity. Next is the structure-activity relationship. The 6-acyl side chain modification gives the semisynthetic penicillins. Electron withdrawing group on the side chain alpha carbon increases acid stability. Bulky side chain near the carbonyl sterically hinders the beta-lactamase attack. Amino group on the side chain, like the ampicillin, enhances the gram-negative permeability and the acid stability. Hydroxyl group on the phenyl ring, as in the amoxicillin, leads to better oral absorption than ampicillin. Urido group, as in the piperacillin, leads to broad gram-negative, including the Pseudomonas. Next is the stability and the degradation. The acidic pH in the stomach, the penicillin G is acid-labile and degrades to inactive penicillin acid. Penicillin V and amoxicillin are acid stable. Basic pH hydrolyzes the beta-lactam ring, resulting into the formation of the penicilloic acid. The beta-lactamase enzyme hydrolyzes the beta-lactam amide bond, leading to the inactive penicilloic acid. Heavy metals and alcohols accelerate the degradation, while heat degrades and hence should be stored in a cool and dry place. The important semisynthetic modifications include acyl side chain replacement, which leads to penicillinase-resistant penicillins, example methicillin and oxacillin. Amino group insertion leads to broad-spectrum aminopenicillins like the ampicillin and the amoxicillin. Carboxyl or urido addition leads to antipseudomonal activity like in the carbonicillin and piperacillin, while the addition of beta-lactamase inhibitors protects against the beta-lactamase. Examples include clavulanic acid, sulbactam, and tazobactam. Next are the beta-lactamase inhibitors. They have a beta-lactam ring but are weak antibacterial agents. They act as suicide substrates, that is, bind irreversibly to beta-lactamase and inactivate it. Clavulanic acid has the oxazolidine ring fused to the beta-lactam. It is from the streptomyces clavulic genus. Sulbactam has the penicillinic acid sulfone, while the tazobactam has the triazole containing penicillin derivative and is more potent than the sulbactam. GPAT or NIPER one-liners and key comparisons are: core nucleus of penicillin is the 6-amino penicillinic acid or the 6-APA. Penicillin G is acid-labile. Why? Because ring strength plus no stabilizing electron or withdrawing group on the side chain. Penicillin V is acid stable due to the phenoxy group, that is the electron withdrawing on the side chain. Methicillin beta-lactamase is resistant due to bulky orthomethoxy groups sterically hinder the enzyme. Aminopenicillins have what side chain? The answer is amino group ampicillin and the p-hydroxy, that is in the amoxicillin. Antipseudomonal penicillin is the piperacillin, carbonicillin, and the ticarcicillin. Beta-lactamase inhibitor examples are clavulanic acid, sulbactam, and the tazobactam. Penicillin mechanism is via the inhibition of the transpeptidase, that is the PBP, and cell wall lysis. MRSA resistance is due to altered PBP2A. Most common penicillin adverse effect is the hypersensitivity, while drug used to prolong the penicillin levels is the probenecid. If you found this content full of knowledge, then do hit the like button, share and subscribe. Stay tuned for next knowledgeable packed video. Do not forget to scan the QR code to download the GPAT Pro app. It is loaded with exciting lectures, pinpoint notes, 20,000 plus MCQs, and much more, and that too at very affordable rate. Professor Gadgil signing off. Jai Hind.